Welcome to Inside Drug Discovery. This video describes a new drug recently approved by the U.S. Food and Drug Administration. The drug is AVMAPKI FAKZYNJA CO-PACK. AVMAPKI FAKZYNJA CO-PACK was approved on May 8, 2025. This was an accelerated approval based on very favorable Phase 2 clinical trial results. AVMAPKI FAKZYNJA CO-PACK is approved for the treatment of recurrent, low-grade, serous ovarian cancer in adults, and we'll place more specifiers on the indication in a later slide. AVMAPKI FAKZYNJA CO-PACK has two APIs. One is avutometinib, and the other is defactinib. The drug is administered orally, with each API being taken separately in different doses and with a different dosing regimen. Often the two APIs would be co-formulated, but that is apparently not feasible in this case. I will speculate on why these might be administered separately once we get into the pharmacokinetics section. The sponsor for the NDA is Verastem. The prescribing information insert of AVMAPKI FAKZYNJA CO-PACK is cited at the bottom of this slide, and this reference was used for much of the information in this video. AVMAPKI FAKZYNJA CO-PACK treats ovarian cancer, more specifically K-RAS mutated recurrent low-grade serous ovarian cancer. You can see at the bottom, K-RAS stands for Kristen rat sarcoma virus. K-RAS mutations are associated with many cancers, including ovarian cancers. The drug is approved for use in adults, and the cancer should have been previously treated by another systemic therapy. The mechanism of action of both APIs is related. Both are kinase inhibitors, though they act on different kinases. Avutometinib is a MEK1 inhibitor with a reported IC50 of 160 nanomolar, according to IUPhar's Guide to Pharmacology. Defactinib shows potency on both FAK and PIK2. According to Kemble, the IC50 is the same for both targets at 0.2 nanomolar. Here are some of the key physicochemical properties of avutometinib. The properties on the right are for the neutral compound, the parent acid, but the drug is administered as its potassium salt. The N-aryl sulfonylurea is reasonably acidic. This is a key idea that we will return to in a moment. Similarly, here are the physicochemical properties of defactinib. Again, the properties on the right are for the free base, but the compound is administered as its hydrochloride salt. So defactinib is a weak base, while avutometinib is a weak acid. Other than these details, the properties on this slide and the previous one are unremarkable for an orally administered compound. Both lie comfortably within oral drug space. On this slide, we have some of the key PK parameters. So these compounds have very different half-lives. Avutometinib has a quite long half-life for a small molecule drug. The steady-state volume of distribution is much higher for defactinib. I suspect some of this difference can be attributed to the positive charge on defactinib at physiological pH. The positive charge boosts association with membrane surfaces and can lead to more extensive distribution. Differences in bioavailability may also be part of the discussion. The bioavailabilities are not reported, but we'll try to infer some trends based on the excretion data in a later slide. Note that defactinib also has a later Tmax than avutometinib. 2 hours is more in line with absorption in the stomach, and a Tmax of 4 hours is more likely in the small intestine. So again, this is a potentially large difference. Based on differences in the half-life and likely sites of absorption, it is more understandable why this drug, the AVMAPKI FAKZYNJA CO-PACK, is provided to patients as 2 separate pills, 1 capsule for avutometinib and 1 tablet for defactinib for dosing. Designing a co-formulation in a single dosage form may not be feasible for 2 drugs that behave quite differently. Let's turn our attention to metabolism and excretion. Avutametinib on the left is cleared hepatically through oxidation as well as non-enzymatic pathways. I'm not sure what the non-enzymatic pathway would be, perhaps conjugation with glutathione at this Michael acceptor. Whatever it is, the rate is quite low because the half-life is long at 29 hours. Excretion of the metabolites is fairly balanced between the feces and the urine. For defactinib on the right, phase 1 metabolism predominates, and excretion is mostly via the feces. Note that the large amount of unchanged drug in the feces implies that defactinib's absorption may be incomplete, and the oral bioavailability, while not reported, may be relatively low. AVMAPKI FAKZYNJA CO-PACK is an oncology drug, so there are several warnings and precautions listed in the prescribing information. Warnings specifically mention ocular toxicities, skin issues, hepatotoxicity, rhabdomyolysis, a condition involving rapid muscle breakdown, and also risks of damage to a fetus. On the next slide, we'll see a number of adverse reactions to the drug, and many of the adverse reactions are directly related to the listed warnings. Here are the adverse reactions. Each of these adverse reactions was observed in at least 25% of patients. I will only highlight a few of these. First, we see evidence of ocular toxicity, which was mentioned on the previous slide. Abnormal liver enzymes in the blood work are a sign of liver toxicity, also mentioned previously. Finally, elevated creatinine phosphokinase, or creatinine kinase, is a biomarker for rhabdomyolysis. That completes this summary of the approval of the new drug application of AVMAPKI FAKZYNJA CO-PACK, which was granted by the U.S. FDA on May 8th of 2025. AVMAPKI FAKZYNJA CO-PACK contains both avutametinib and defactinib as its active pharmaceutical ingredients and is approved for the treatment of recurrent low-grade serous ovarian cancer in adults. Thank you for watching this video. Please consider liking the video, subscribing to the channel, making a comment, or leaving a question. Take care.